Yes, I did that.
Yeah, okay. Hey, don't come back. Yeah. Juniors, share it in your groups, okay? The link.
We go live exactly at 8:30, right?
Mm.
Okay.
No, I think people can hear us. Sorry.
So should I start?
Yes, please go ahead.
So I'll start off, right?
Yeah, you can begin. So, hello everyone, whoever is there. Welcome to the Science Month. And today is our first talk. And yeah, it hasn't started yet. We are going to begin. You're just in time.
Yeah.
And we'll wait for a bit because we'll let a bit more viewers come in, and then I'll pass it on to Adrija, who will tell you about our speaker and our event.
Okay. So I'll start. Hello, everyone, and welcome to the first talk of the Science Month, which is organized by Campus Radio and the Science Club. Today's talk will be delivered by Sushma Ambekar, which is titled How Did I Become a Malaria Researcher? Sushma started off by pursuing an engineering bachelor's degree in biotechnology from BMS College, Bangalore, where she realized that her inherent love for molecular biology and doing experiments in the laboratory resided. And this is exactly when she decided to pursue a PhD and is currently a fourth year graduate student pursuing her PhD in the field of Plasmodium biology, or more particularly, as she says, it's on the field of molecular parasitology. Since last year, she has been extensively associated with SciComm and has been volunteering in all of the events involving talking to people about her research and science in general. We welcome you, Sushma, and hope that our audience have a wonderful time hearing your talk and your field of expertise. A slight request to everyone, all of your doubts can be directed to the comments of the YouTube Live, and we'll be taking them at the end of the talk. Thank you.
Thank you very much. I think to begin off, I should thank the ICER Campus Radio team. I think you guys are doing a wonderful job. If at any point you have difficulties hearing me due to internet issues or microphone, just let me know in the chat and I can quickly reconnect or turn off my video. But again, this is a great chance to speak to you all. I'm very glad that you're joining in, even though it's evening, almost night in India. So thank you for doing that. For today, I have a very short story planned of my very short journey in the field of biotechnology and molecular biology. I hope you will find this interesting. I hope you'll be able to connect with this, and we can talk more after my talk is over. So let me go ahead and share my screen. Okay. Can someone confirm that it's seen? That's okay.
Yes, we can see it.
Okay, perfect. Thank you. So again, thanks to everyone who is joining in. I would like to again introduce myself. I am Sushma Ambekar, and I'm a PhD candidate at Iowa State University in the US. And today I will be talking to you about how the last chapter of my biology textbook in my +1 and 2 led me to become a malaria researcher today. A quick timeline of what's been happening for the last 7 to 8 years. So I started off at a pre-university college, and I have always had an interest for biology. So naturally, I took up biology subjects in my pre-university college as well. And my textbook had a lot of different lessons, as I'm sure you've seen. We had a lot of anatomy, we had a lot of botany stuff, but the last chapter, and this was fairly, not a very new subject at that time, but fairly unknown subject at that time. And this was a lesson called Introduction to Biotechnology. And until then, I had not heard about this. I had no idea what this was, but it intrigued me very much. And that's when I got started. That's when I learned about the importance of biotechnology in today's world, the wonders of genetic engineering, and all that it has to offer. I went on from my pre-university college to do a bachelor's in engineering, but in biotechnology from 2013 to 2017. In the 4 years that I did my BE, I also did multiple internships during this time, which I think really helped me get a lot of experience and perspective on what this field really is. Some of the internships that I should really mention are my internships in Skanda Life Sciences, which helped me pick up my skills in cell culture. I also did an internship in Coca-Cola, where I learned bioprocess engineering and everything that goes into scaling up these processes. But I think the majority of my experience that helped me get into my PhD program came from this startup called String Bio. I will talk about it more in the next slides, and I could not be more grateful for this company for helping me build my career. So following these, I graduated from bachelor's and decided to do a PhD straight away. And that's when I got into Iowa State University. I've been here since 2017. So I started at the PhD program at the Molecular, Cellular, and Developmental Biology program. It's an interdepartmental program which will really allow you to explore more than 10, 15 departments, in fact. And there are a wide range of research topics that were open to me. And that's how I ended up choosing malaria research, and that's what I'm doing today. So going back to what I said, my major internships for over a year and a half during my last year of engineering was in this company called String Bio. So this is a startup in Bangalore at the Bangalore Bioinnovation Center. And Bangalore Bioinnovation Center really has a lot of startups. As of today, I think they host about 20 to 30 different startups, and they're always open to interns. And I have always been recommending this place to anyone who is trying to find an internship. What String basically does is they make use of these bacteria called methanotrophs. Methanotrophs are bacteria that use methane instead of carbon dioxide or oxygen to live and survive. But what they do is they make use of these methanotrophs and genetically engineer them to make products that they want. They make it go down the metabolic pathway that they want. And in this way, they are trying to make biodegradable plastic made with these methanotrophs. So there for over a year and a half, I was closely related with the molecular biology and the genetic engineering of these bacteria. I did not work a lot with the bioreactor side of it, but all of the cloning techniques that I learned while at String helped me to get into this PhD program in the first place. And that amount of lab experience was extremely invaluable at that time. So I really owe it all to String for helping me even get into the basics of molecular biology and genetic engineering. And this is where I got really, really interested into this whole field. Following that, as I said, I came here to Iowa State University, and here we have this great opportunity where we don't have to choose a professor right away, but we can spend the first 6 to 8 months rotating in different labs. So I had a chance to rotate in 3 different labs. The first was a lab by Dr. Chris Tuggle, where they work with humanized SCID pigs. So as you know, SCID is severe combined immunodeficiency. So these pigs are already immunodeficient, which means they don't have an immune system. So they work as great model systems to understand the immune system of humans. And as you know, humans and pigs are very close. So I got a chance to work actually with these SCID pigs and also do a lot of cloning while I did this for 2 months. The second one was where I am currently. I work with Dr. Gunnar Meyer to study the nuclear pore proteins in the malaria parasite, Plasmodium. And here I was able to work with mice models. So I got a lot of experience working with rodents, which I previously hadn't been able to do. And of course, I was also able to work with blood culture because Plasmodium parasites grow in blood, and I will again tell you more about this later. The third one was, again, very different. I worked with Dr. Walter Moss, where they study RNA sequence, structure, and functions of different types of RNAs, including non-coding RNAs. And here I was able to pick up more of cell culture techniques. And I think all these 3 put together really helped me decide what I want to do. And regardless of what lab I joined, it helped me pick up a lot of techniques. So this idea of lab rotations was really helpful, and that's how I ended up at my lab where I am for the last 3 years. So let me tell you a little bit about my research. I'm sure most of you have heard about the disease malaria. Malaria affects millions of people each year and kills about 500,000 people every year. In fact, as I speak, as I just get through the slide, there is a child in Africa probably somewhere dying of malaria. That is the severity of malaria. And of course, we have drugs for malaria, but we don't have an affordable and easily accessible vaccine yet. There have been some vaccines that have been developed, but none of them have proven to be very efficient. And this is why it's very necessary to understand the organism that causes this in the first place, which is Plasmodium. So what I'm showing here is how how humans are affected with malaria. So mosquito carries this Plasmodium parasite, and when a mosquito bites a human, these Plasmodium parasites travel to the liver, where they grow and differentiate, and they're released as merozoites into the blood of the human. In the red blood cell, they go through an asexual reproduction cycle, where they go through ring stage, trophozoites, and schizon stage. Basically, one single merozoite becomes tens of these schizons, and the red blood cell ruptures, releases these individual parasites, and they go on to infect more red blood cells. So this is when the symptoms of malaria are seen: fever, shivering, and all of the other mentioned symptoms. This is the stage where most of the drugs are directed towards, and this is also the cycle that we are currently interested in studying. As I said, this malaria is caused by the protozoan parasite Plasmodium, and Plasmodium is actually a unicellular eukaryote, which means it's just a single cell organism, and it has a membrane-bound nucleus like our cells, and it carries out all the functions with the DNA in the nucleus, just like any other eukaryote. Why I'm telling you this is because that's what we are interested in studying. Now, as we know, the nucleus is the house of the DNA. It holds everything in itself and it guards the DNA. But DNA needs to get out into the cytoplasm as mRNA to be made into proteins. So the nucleus, which is bound by the nuclear membrane, actually has tiny pores in it, hundreds of tiny pores, which help any kind of material, biomolecules, move in and out of the nucleus. Now, these nuclear pores are not just holes in the nuclear membrane, but they are guarded by these complexes called nuclear pore complexes. Now, this is what we study, and to make it easier, I'll call this NPC, nuclear pore complex. The nuclear pore complex is in turn made up of about 30 different proteins, and this forms a huge megadalton assembly in pores. So these are basically the guardians of the nucleus. They decide what goes in and out of the nucleus. Now, why are we so interested in studying this? 80% of the Plasmodium nuclear pore proteins remain unknown. So there are about 30, and we know this 4 or 5 as of today. And considering that this Plasmodium parasite causes so much trouble to humankind today, it's really important for us to understand these proteins, identify these proteins in order to understand their functions. Because what we're talking about is the nuclear pore membrane, it closely interacts with the DNA that is inside and is basically telling how much of the DNA should be made into protein, what should be let outside. So if we're able to identify these proteins and see how they are carrying out these functions, we could potentially stop this and maybe make drugs or vaccines against this parasite. So how are we doing this? I wanted to talk about this because this is a fascinating method that has been introduced in the last 5-6 years. This is called as proximity labeling. Basically, think of it this way. This is an easier analogy. It's like fishing. So you have a bait, and then when you put it into water, the fish is attracted towards it, and you can catch the fish with it and see what you got. This is exactly what we're doing. We use something called a bait protein, something that we already know. And as I said, we know 5 of them. So we have quite a bunch of bait proteins. And then we attach this enzyme called BIR-A, that is biotin ligase. And what this does is, in the presence of biotin, the biotin ligase will biotinylate the proteins, which means it will add these biotin molecules on top of proteins that are close by this biotin protein, exactly like fishing. It catches the proteins that are nearby, and we're able to identify these using streptavidin beads. Now, in this field, the streptavidin and biotin interaction is so strong that it's basically like a magnet. Like when we put streptavidin, it's able to capture all these proteins with biotin in it. And then we're able to identify these using mass spectrometry and see what we have in hand. So this way, we're trying to identify the unknown proteins that are in this parasite Plasmodium. So that is about my research. Apart from that, I have also become very closely involved in SciComm activities in my university. So I recently took part in this Science Center event that happened just in a place very close to where I live, where we were able to interact with school children, and we were able to take a microscope there, show them different types of cells, and they were really fascinated with it. What was the challenging part for us was explaining these scientific terms in very, very simple words. There were kids from the age of 5 to 15, so it was an absolute challenge, and their parents were equally interested in this as well. So it was fun. We tried to make this as engaging for them as possible. We took a lot of crayons and craft stuff so they could make viruses with tiny pom-pom balls. And it was a lot of fun. And of course, this was pre-COVID, and since then we haven't had a chance to have such events. But following that, I've been trying to have such kind of talks with undergrads in my college and in some other colleges as well. I try to keep a blog. Apart from my lab time, I try to read and write about things other than my research as well. And the other thing I'm very interested in is the 3-minute thesis. I'm not sure how many of you have heard about it, but you haven't, you should definitely look it up. It's basically explaining your research to a non-experienced audience to a non-specialized audience in 3 minutes. And you should not bring up a lot of jargon, but you should not water down your research as well. So the challenge is explaining your research in 3 minutes, and that's all you get in one slide. So I've taken part in a couple of these 3MT competitions at the university level, and it has been amazing to think of my research in a very broad sense, in a way that anyone would understand. So at this point, I'm sure you're thinking, all this information is great, but of course you're going to have a lot of questions as you go through undergrad and even ahead. And where can I find this information, and where can I find people that I want to talk to at the time I want? So I have a couple of things that I have learned in the past year or two, so I'm going to share that with you. Science Twitter is a very big thing right now. A lot of scientists, a lot of researchers from everywhere across the globe are on Twitter. So if you're not on Twitter and you're thinking about it, you're not sure, I would say definitely join Twitter. You can connect with literally any professor, any postdoc, any PhD student on Twitter. And most of them are always happy to help you. They even advertise PhD positions on Twitter. They advertise the conferences on Twitter and open days, which you should definitely attend. So if you're looking for a place to connect and network and find more information, Science Twitter is definitely the place. And of course, this is pretty obvious. LinkedIn, I'm sure a lot of you are already on LinkedIn. You can find internships and jobs, and you can find alumni of your college as well, where you can ask questions because they might be in different places. And thirdly, thanks to this group, I am here. So there is a group called International Scholars in the US, and they provide they basically connect people, researchers in the US. And recently we created a channel for prospective students. So if you want, you could join that group, and you could ask more than 700 people who are there for help, and I'm sure they would be glad to help you. And this is another thing that I recently came across. I am not sure if you are familiar with this or if you're already there on this, but there is a group on Facebook called STEM Peers. And people post all sorts of questions about vacancies, about colleges and everything. So you can check this out as well. With that, I think I have covered what all I had in mind. But I would like to really thank the ICER Kolkata campus radio team, and especially Sumit, for reaching out to me via this same Slack channel that I mentioned in my previous slide. I want to thank my professor Gunnar Meyer for giving me this opportunity for the last 3 years to do this PhD with him and introduce me to this amazing field. And then there is another professor here called Dr. Jospek, who also does research on malaria. So I should really thank him because they helped me with a lot of my experiments. And of course, my college in Iowa State University for helping me give this atmosphere to carry out this research. So if you have questions, you can reach out to me. You can take down my email. You can reach out to me on Twitter or LinkedIn. I would be happy to help literally anyone with any questions regarding anything about colleges, about how to find your field that you're interested in, internships, anything. I know information that I have gathered from my journey, and I have friends who have done the same as well. So I would be happy to connect you with anyone. But thank you very much for listening to me, and I can take as many questions as you have. I don't know how much time I took. Okay, 22 minutes, not bad. So over to you. Thank you.
Thank you so much, Sushma. That was a really interesting talk, and especially the other than science, how to make connections and how to do science communication, and all that is an equally important part of being a researcher in academia. And we really appreciated that you shared that information with us. So we do have some questions that I'll begin asking, and anyone else who's watching can actually go on and put their questions in the chat box. So the first question is by Omkar Sahu. Can you kindly once again explain how you identify the malarial NPC proteins?
Oh, yeah, sure. Thank you, Omkar. Let me go back to my slides so it's easier. Yeah. So what we're doing basically is, you see, the green that I've mentioned here are the proteins we already know. And of course, there's different types of NPC proteins. So we already know these proteins. So we're using them as bait. So the protein is literally attached to this enzyme called the biotin ligase. And when this is attached, we make a plasmid from this and insert this into the Plasmodium parasite. And this replaces the protein that is already there. So now the Plasmodium has a copy of the protein which is attached with the biotin ligase. So when we grow these Plasmodium parasites in mice, there is already biotin in the blood, but also if we do a cell culture, we add biotin to it. So in the presence of this biotin ligase enzyme, which is along with the protein, is actually adding a biotin tag to every protein that is around. So if you look at this is a pretty tight-knit complex, right? So if you add a biotin ligase to any of these proteins, it's going to biotinylate all the proteins close to it. So what we're trying to do is we're trying to catch the proteins in the complex because they're all very closely put together in a complex. And then we collect these Plasmodium parasites which have been biotinylated and then extract the protein from it. And then we're able to identify them using mass spec. So that's how it goes. Let me know if you understood that.
Great. So, Omkar, if you have any further doubts, you can actually ask, or you can email her. I'm sure she'll be glad to take your question and answer. And so the next question is from Adrija. What is the difference between a proximal and non-proximal protein?
It's exactly as the name suggests. So proximal proteins are proteins that are in close contact within 10 to 25 nanometers in the protein complex, and non-proximal proteins are anything that are far away. So in the nuclear pore complex, all of these are pretty close, and then there are some proteins apart from the nuclear pore complex in the membrane that are considered non-proximal. So it's literally as the name suggests, they're close by their proximal or else they're not.
Great. So another question is, did you participate in IGEM? I guess IGEM stands for International Genetically Engineered Machine. That's a global competition of synthetic biology. So did you participate? And if you did, kindly let us know your experience.
Thank you for that question. So I unfortunately became aware of IGEM pretty late in my engineering. I think by then I was past the chance where I could apply to it. But there is another competition called BioMod. I'm not sure if you're aware of that. But BioMod is also a biological modeling competition, sort of like IGEM, that some of my friends took part in. I took part in this competition called the Biomimicry Challenge, which I think happens every year. It's an international challenge where you have to come up with a product design, but inspired by biomimicry. So if you're not familiar with biomimicry, it is basically taking inspiration from nature. So they make some materials mimicking the beehive structure because it's very strong. And that is basically biomimicry. So I did take part in that, but not in IGEM. I wish I had. So if you ever have a chance, please do.
Great. BioMod is something that I personally didn't know of, but that sounds equally interesting.
Yeah.
And, yeah. Another question is by Alekhya, and they're asking, do you think this research can be later studied further for therapeutic intervention? If yes, then how?
Oh, thank you. That's a great question, actually, Alekhya. Thanks for that. Yes, this research can be applied for therapeutic intervention. So right now for cancer as well, there are a lot of drugs that basically stop this process of these complexes getting together. So when there are a lot of proteins, like 30 proteins, there should be a process in the cell where they're forming a complex, right? So there are a lot of drugs that are stopping this process in cancer, and the same can be applied to Plasmodium as well. These are basically called anti-mitotic drugs because this whole process of making new things happens during mitosis. So this stops the cell division and basically can stop the Plasmodium parasite from dividing and making more. So I would say, from as far as I know right now, that would be a great therapeutic way to put this to application.
Yeah, thank you for your answer. And I guess the one who asked the question is satisfied. If not, then you can always go on and ask again. Next question is by Tanishta. Can you please share your experience of finding internships? How does it work and what are your experiences?
Yeah, actually, yes, I would love to talk about that. So as I mentioned, I did 3 different internships and I think I found them in 3 different ways, which is a good example for this. So my first internship was in Skanda Life Sciences. I literally Googled biotechnology companies near me on Google, and it came up with a list of companies. And this was following my second year of engineering, so I did not have a lot of contacts then. So, and there was this company that was I think 2 or 3 kilometers away from my house, and I thought, oh great, I can just go work here. So I just went. I literally walked into their company and spoke to them, and they said that they would. It was basically a training experience, so I actually paid for it. So I think I paid 5,000 or 6,000 rupees. But looking back, it was totally worth it. And they let me work there for two months, and they gave me a lot of experience, and they taught me how to do stuff. So I think that was really a training experience and me helping the company. My second experience was in Coca-Cola. So that I spoke to professors in my department. I started like a month before summer. Actually, I did that in winter. So I started a month before my winter break, and I asked every professor in my department if they knew any alumni that is in a company right now and working. And one of them did. So they knew someone who had passed out 5 years earlier or 4 or 5 years before. And they put me through and they made me contact him. And he was able to get me a very short internship at Coca-Cola. Then the third one, String Bio, actually came by chance. So they were a relatively new startup then. They were just 2 years or 3 years into being, and they came to our college to actually spread the word about their company and actually pick up interns because they wanted someone to help them with their work. So that's how they did a short interview kind of thing. I wouldn't say interview. They actually gave us tasks to do and chose the people who did well at it. So that's how I found String. But that was actually for 2 months. And then we spoke to them and asked them if we could do it for a longer time. And they said yes. So I think the third one, they came to the university. So, I mean, look out for companies like that if they are contacting your department or even flyers in your department. But other ways you can find internships. There's Internshala, which I've heard is also good. The other thing I did not do is directly talk to my alumni. So if you know, if there is a group or if there is an email which you can contact your alumni with, do that because they're obviously in companies or in different colleges now. So that's another way you can find internships.
Great. Thank you so much for the insight. And we have another question from Priyanuj, which is contacting research groups through Twitter considered informal or is it equivalent to emailing? What are your views on this?
Thank you, Priyanuj. Twitter has become kind of an informal space. LinkedIn is, on the other hand, more formal. But as far as I have seen, Twitter is very informal. Let me give you my experience from this. So I came across this professor whose research I was very interested in. And I may or may not join their lab, but I was interested in getting to know them. So I tried to find them on Twitter and they were there. So I just followed them and they did not follow me. Of course, professors have so many more things to do. But I sent them a message and it was pretty informal. I sent them a message saying that I have read about their research and their lab is doing really good stuff. And I would like to talk to them about their research someday. And I would like to connect with them basically. And they followed me back, and ever since then we've actually been running into each other many more times. So it is pretty informal. Feel free to you can follow. You don't have to request a follow on Twitter. So that's the other great thing. Most of the professors have their profiles open, so nobody keeps it private. So you can just follow them and then send them a message. Shoot your shot, and if they reply, and you can connect with them. So it's a great way to not be formal, but still show that you're really interested in connecting with them.
Cool. That was really interesting. And so is the next question, which is a really fascinating question by Ame. And they're asking, what are your views on the current research on gene drive, the one where eliminating entire malaria-causing species is being considered? So, yeah.
Thank you. I have read about this and seen a couple of videos on this, and I think there are two approaches to do this. I thought that one way was to stop these Anopheles mosquitoes, which are the Plasmodium mosquitoes, to make them stop spreading this Plasmodium species in the first place, and second is completely eliminating them. I'm not so sure about completely eliminating them, because mosquitoes do more than just carrying these diseases. Although it wouldn't be too bad to not have a bunch of these species, we would still have the other mosquito species. But what we don't know is the effect that would have on our environment. We can only hypothesize and say probably nothing is going to happen, or come up with proofs that nothing is going to happen. So I should probably read more into it, but until we have further proof that this will not have a negative impact on our environment, I wouldn't be very sure of doing this on a larger scale.
Yeah, we can never be sure until we actually do something. So thank you for your answer. And the next question is by Deepta Tanudas, and they're asking, what's your view on nucleopore protein being used as the next-gen single-molecule analyzer?
I'm actually not very sure about this. So I'm not sure what they mean by next-gen single-molecule analyzer. Are you saying as a diagnostic method, or I'm not quite sure about this. I'm not sure I get the question.
Yeah, so maybe Deepta Tanu, you can try to clarify, or you can always email her if you have any more questions.
Yeah, I would be happy to talk about it if you can email me.
Another question just popped in, which is by Alekia, and they say, it's a silly question, but what are your views on vaccines for mosquitoes?
Oh, there is no such thing as a silly question, so don't call it a silly question. Vaccines for mosquitoes. Don't you think it would be extremely difficult to vaccinate mosquitoes? I mean, I think gene drive is probably the easiest approach we've had so far, because the mosquitoes, they will try to reproduce and they will make more mosquitoes that can't carry the parasite anymore. But the one previous approach that was used to get rid of the mosquitoes was spraying these chemicals that got rid of these mosquitoes, like DDT, if I'm not wrong. In previous in history, that's what we've done. But vaccinating mosquitoes might be a little difficult.
Yeah, so we are waiting for more questions, and till we get more questions, we can have a discussion about your PhD experience, I guess. So you can tell me about how do you feel? How is a PhD compared to a bachelor or master's degree, and what kind of work-life balance are you able to achieve during a PhD? Because that's the complaint of most academics, that they're not able to maintain a work-life balance. What are your views on this?
Okay, thank you. That's a good question, actually. I should say I have really enjoyed my 4 years of PhD. There have been times where I've felt pressured, but again, that was when I had deadlines or when there was experiment to be completed at a particular time. But otherwise, when I look at it, when I look back, I think I have really enjoyed my PhD experience. How has it been? So I can't really compare with masters personally because I directly got into PhD. But I know I know my friends who've done a master's who joined exactly when I joined my PhD. And I would, if I had to go back and choose again, I would choose PhD again. That's because of the amount of research experience that I'm getting, not just the actual experiments that I'm doing, but the people I'm able to interact with and the mistakes I'm making. I have made a lot of mistakes in my experiments and I have learned from them again. PhD gives you a chance to really design your own experiments, make mistakes, and then correct them and design the experiments again. And that kind of experience is something you won't get anywhere else. Because you have these 4 to 6 years to experiment all this and learn. And this is what you will apply for the rest of your life, whether you continue in academia or join an industry. The actual difference between masters and PhD. In masters, you will take 8 or 10 classes or probably more than that. You might do a research assistantship on the side, but you will not really get involved with the project. Like they will assign work to you, you will do that, and you'll probably get a couple, 1 or 2 second or third author papers, and you will get out. And then you can do whatever you want. But in PhD, you really start off from scratch. Of course, there are side projects that you do with other people where you're doing bits and bits of experiments, but you have your thesis where you're starting from the beginning and you are designing the experiments. So that gives you need to look up protocols yourself. You need to find the papers. You will have 10 different protocols and 10 different papers. You need to apply logic and reasoning and come up with what the actual protocol is that you need and which is good for your experiment. So that way, I think PhD has been great. And of course, you have a lot more time to actually figure out what you want to do next. Because there are choices. You can stay in the same field that you do your PhD in, or you can shift to a slightly different field. So you get to connect with so many people in 4 to 5 years, and I think that has been a great advantage for me. So yeah, I would, if I had to go back, I would do PhD. If I had to choose again, I would do PhD again. Oh, and sorry, I forgot to answer your work-life balance question. I think I've been pretty good at maintaining work-life balance. I try to take most of my Saturday, Sundays off. Of course, because I have mice, I need to come in sometimes and take care of my mice for an hour or 2. But then I go back. I try to have other hobbies. I have a big garden at home. It's like my family away from home. So I do gardening in my spare time. I bake a lot, and then I read a lot of other books other than science books. So I think having other hobbies is equally important, and making friends is also very important in your PhD. So with that, you can definitely maintain a work-life balance.
Great. Thank you for the insights, and that also inspires all of us to go on and get a PhD. There are a few more questions. Dita Tanudas has clarified his question by saying that application in protein sequencing where we can use the transport selectivity of the nucleopore protein based on interactions along the radius of the pore to link molecules. So like a filter, I guess. So thank you for that. This actually makes it much clearer. So I don't think we could really use this in DNA sequencing, but we could. So these nuclear pore proteins are also known to interact with chromatin. So they interact with euchromatin, if I'm not wrong. Basically, a chromatin that actively makes more
the mRNA. So if there are some of these genes that are unknown, we could potentially use techniques such as chip, chromatin immunoprecipitation, along with these proteins to identify these, or we could also study to understand the modifications on chromatin. So chromatin modifications like phosphorylation or acetylation dictates what happens to the gene, whether it is transcribed or not. So we can use these nuclear pore proteins because they interact with such things to understand the modifications on chromatin and therefore understand the state of the gene. So that's one thing that we could do. But you bring up a great point here when you say transport selectivity of the nuclear pore proteins. So there are a group of these nuclear pore proteins called F genomes, and they are the main thing that let biomolecules even pass through. So they have phenylalanine glycine repeats, and they actually form a mesh in the center of the channel, and that's where cargo passes through. So imagine if you're able to make drugs where you can block these FG, whatever is helping these FGs interact with the cargo, you can basically block the transport. So you are able to do a lot more in terms of therapeutic applications to this.
That was pretty insightful, and I guess Pratanu, you were satisfied, or you can always reach out to her with this email. Next question is by Manas, and they're asking, is there some research going on which is targeting something other than NPC, like blocking transcription or translation, etc.?
Yeah, there is. So in Plasmodium, because as I said, we don't have a vaccine yet, there is a wide range of research that is going on. If you're ever considering this is an amazing field. And because we don't have a vaccine, it's an up and coming field. So yes, they are doing, there are a group of transcription factors in Plasmodium called AP2I and AP2Z, and AP2 basically the family of AP2 transcription factors. So there are multiple research groups studying these transcription factors and trying to understand what they're interacting with so they could block them. Right now, I don't think there is something that is already known which can target these, but they're trying to come up with the mechanisms first, so then we could target it.
That was really cool. And next question is by a first-year student, a fresher, and they're asking, according to you, is it possible for a student to balance both physics and biology? And they are a BS student, a BS MS student. So what are your views on that?
You can absolutely do that. There is a great field called biophysics, and it studies, because biology, chemistry, and physics are very close knit, right? I'm sure you'll agree with that. Cells in our body also undergo certain stresses, and there is a lot of energy exchange. I am not very great in physics, as you can already tell, but there are a lot of different research topics that beautifully merge biology and physics. So if you want to stay in the field that is between these two, biophysics would be your place. There are a couple of labs studying how cancer cells divide with respect to the stress that they have and what are the energetics of division and all of that. So you should definitely look it up, and biophysics would be your research area.
Yeah, so I hope, Swarnendu, that answers your question. And the last question, I guess, is the same student who had asked, what are the possible options a BS MS graduate student can pursue apart from PhD or academia?
Yeah, yes, apart from academia. So there are 2-3 different ways you could go into, I guess. Following your BS MS, you could, of course, apply to biotech companies in India. There are so many. My knowledge of other cities is pretty limited because I did not explore much. I should have. But in Bangalore, you have companies like Biocon or Syngene or Novozymes. So many pharmaceutical companies that are studying these different drugs or any other pharmaceutical approaches, basically. You could directly apply to them. I know for a fact they really are interested in BSMS students because they have actually picked up the necessary lab skills for their experiments and their projects. The other way you could go is you could become a research fellow without a PhD track. I think you could write the CSIR exam and you could become a junior research fellow at any of the top research institutes that we have in India. You could also apply just as a research scientist or a research associate to any of the labs abroad. A lot of them take this. You don't necessarily have to do a PhD to work in a lab, even in the US. I'm not sure about Europe, but at least in the US, I know you can just come in as a research scientist. If you have experience in that field, if there is a lab looking for your expertise, they will definitely take you. That is all that comes to my mind right now, but there are a bunch of different options other than just doing a PhD.
All right, so there's one guy who's constantly messaging me. He's Omkar, and he has this one question. So he's asking, how actually did the last chapter actually help you to zero in on malaria?
Thank you, Omkar. I think you're actually helping us wrap this conversation up together. So I did not know what biotechnology was until I was introduced to that lesson in my biology textbook. My friends or family had not heard about this before and were not very aware. So I think that was really my first introduction into the field very closely. And that's what helped me become interested in molecular biology, because I felt cloning was something magical and amazing, and you could do literally anything. You could clone, you could do genetic engineering to any organism and make study whatever you want. So that's how that continued. And because of that, I even applied to PhD programs, and that's how I ended up with where I am today. My research on a daily basis involves a lot of cloning. I need to basically design a lot of cloning experiments, design primers, come up with the cloning strategy. So I think that interest that started from the last chapter of my textbook has carried on until today. Without that, I probably would have done something else that I was interested in, but I wouldn't have been introduced to this. So that's how the last chapter led me to become a malaria researcher.
All right, so I guess, Omkar, your curiosity is satisfied, and it also brings to a fitting conclusion to our entire program. And I guess that's about it. And I guess you guys liked our program, this wonderful talk and this wonderful discussion. And if you guys have any more questions, queries, go forward it to her. Her email is visible on the screen. And if you have any feedback on the event, if you want to say something that you could not in the chat, then you are always welcome to reach out to us at our email address. And everyone is very thankful for the event, Sushma, and they loved it. That's what they say in the chats. And I guess so we'll be ending the stream now. Thank you guys so much for attending. Without you, it would not have been possible. And
Thank you. Thanks again for inviting me. And if anyone has any questions, if you're not able to reach out to me, you could always ask Sumit, and Sumit, you could always direct them towards me. I'd be happy to help you in any way I can. Thank you.
Thank you all.